Okay, we're back. Biology 160. And again, worksheet 6, cell structure and function. We're on number 7, and I had just more or less finished discussing chromatin strands. And we had a picture that looked sort of something like this, where we left off. And this was our DNA, and these were associated proteins that are bound to the DNA, and the two things together, the DNA with the proteins, equals chromatin. And I suggested that you go find some images in Google or elsewhere for chromatin and histone proteins. So I found a couple illustrations in another book that I will show you quickly. So this picture shows, here is the DNA. Don't worry too much about the structure, but this is the double helix. You've probably seen this. So this is the DNA, and DNA gets condensed, wrapped up. It's a very long, thin molecule, and inside the nucleus it gets coiled up around these proteins, and these are histones, the most common of these DNA-associated proteins. And there's 4 of them. They form sort of a complex, and then the DNA wraps around them, and then it goes off and wraps around another one, wraps around some more, and that's how you kind of coil up the DNA. And if you look at this underneath an electron microscope, you can see the little histones, which they're referred to as a little nucleosome. Don't worry about that term. I'm not going to ask you about it. But at any rate, that's chromatin. It has the little black protein dots and the DNA wrapped around it. And then if you want to just kind of continue this condensation, this gets wrapped around again and again and again, and then it gets coiled up into these chromosomes that we'll look at later when we look at cell division, mitosis, and meiosis, and we start to draw them as these X's. So at any rate, this was a picture of chromatin and the DNA-associated proteins, histones being the most common. Just sort of FYI for completeness, here's another picture out of a different book, same idea. This shows perhaps a little more detail of the histones. This is the DNA, it just wraps around the histones. All we're really asking you to know in this class is that the DNA plus all the protein makes up a chromatin strand. So chromatin is DNA plus protein. And I think that's enough of chromatin strands. The next thing that I'd like to do is to go through on our list. It is ribosomes. Find my pen here. So let's go through ribosomes. And ribosomes are very small organelles. I mean, all of this is microscopic, but they're relatively small compared to other organelles. Small organelles. And they have no membrane around them. So it just turns out that these are non-membrane bound. Okay, other organelles like the nucleus, the mitochondria, lysosomes, etc., they have cell membrane or phospholipid membrane around them, but ribosomes do not. And their claim to fame, something that you need to know, the function of ribosomes is that this is where amino acids are joined together to make proteins. So amino acids are joined at the ribosome. The ribosome is involved with joining amino acids to make protein, because protein is made up of amino acids, and you've been through those biological molecules, so that should sound familiar. And this is the business of protein synthesis. The synthesis of proteins is making proteins. So ribosomes are essential for that. And as I mentioned earlier, there is a large and a small subunit. And you remember that those subunits were assembled in the nucleolus. We previously talked about that. So a large and small subunit. And I would like to show you a picture out of a different textbook. This is out of an old A&P book. Hopefully it shows up here. Okay, I'm going to play with the zoom for just a minute, see if I can back out a little bit. And I guess that's about it. Okay, so it's a busy figure, don't panic. All I want to do is point out ribosomes. So let's get oriented in the figure. This is the cytoplasm of the cell. Here is the nucleus. You recognize the double membrane around it. Each one of those is a phospholipid bilayer. And there are pores within this nuclear envelope that allow things to move back and forth into the nucleus and out of the nucleus. And the pink dot region is the nucleolus, inside the nucleus. So this is the nucleus, and this is some of that DNA chromatin. Remember we said that DNA was contained in the nucleus? Here's the DNA inside the nucleus. And we're talking about ribosomes. So ribosomes are made up, here are what they are showing as ribosomes. These, don't worry about number 4 down here for the moment. But this is a small subunit, a small ribosomal subunit, and a large ribosomal subunit. Now those subunits were assembled back in the nucleolus. We've already talked about that. So what is the ribosome? These small subunits, what are they made up of? Well, they're made up of proteins. And here are some of the proteins. These are ribosomal proteins that are made out in the cytoplasm. And then those proteins move from the cytoplasm back into the nucleus. And then they move into the nucleolus. The other thing that's in the nucleus, in addition to these proteins, these ribosomal proteins, is RNA. And this is rRNA, which we'll talk about later. Don't worry about that too much. It's just RNA. And the RNA gets together with the proteins that are present. And the RNA plus the proteins get folded up into this gamish and then exported out of the nucleolus into the nucleoplasm, back in the nucleus. This is the RNA in red, and the little ribosomal proteins, the little blue dots, and those get moved out, and that's what this is. So this, each subunit, the large subunit and the small subunit, are made of RNA and protein. Okay, so now we know what the ribosomal subunits are made up of. Okay, I'm going to get rid of the picture and go back to our notes. So each of these subunits are made up of proteins plus RNA. It turns out that this is ribosomal RNA, so sometimes a little R is put in front of it just to denote that it's ribosomal RNA. And that's what makes up the ribosome. Okay, good. So now, these ribosomes are going to be making proteins. And you find the ribosomes either sort of free-floating in the cytoplasm, or those ribosomes can be bound to the endoplasmic reticulum, which we'll talk about next. And nobody likes to write out endoplasmic reticulum very much, so it oftentimes gets abbreviated as the ER. As long as everybody knows what we're talking about, after we spelled it out once, we can kind of abbreviate it. And we have two types of ER. We have rough ER, or RER, or we have smooth ER, or SER. Rough endoplasmic reticulum, smooth endoplasmic reticulum, they do different things. They have different functions. So I'm going to, let's go off now and, oops, I'm sorry, I was out of view on that. So the rough ER and the smooth ER. So let's talk about the rough endoplasmic reticulum. I'll go rough ER just to speed things along, going down the list of your worksheet packet. So a couple of things to note about the endoplasmic reticulum, the rough endoplasmic reticulum. I'll show you some pictures in a minute and we'll discuss its function. But it's a series of, I don't know, let's say flattened membranes. So it's a membranous structure inside the cell, in the cytoplasm. It's an organelle. And it's close to the nucleus. And this is the site of protein synthesis. So this is, let's say this is the site of protein synthesis for proteins that will be exported out of the cell. And then they would go into the blood or into these other extracellular fluids. But they're going to be, all proteins are made in cells. All proteins are made with ribosomes, and those that are going to be moved out of the cell are synthesized in the rough ER. So another function of the rough ER is that when the proteins are made, the rough ER is responsible for folding and modifying proteins. Proteins have a very specific shape, so you have to build that shape. And that gives them a specific structure, and hopefully you recognize the relationship between structure and function. If it has a specific structure, that gives it a specific function. So the shape of the protein is everything. And that shape gets built in part the rough ER. So let me find that other picture. And this is what you have in your book. So this is the OpenStax book. And this is the figure. It's 313 on page 67, at least in the edition that I have. So, a couple of things. Let's get oriented. There's the nucleus. And all of this is the blue shaded stuff. Forget about the orange dots for just a moment. But the blue and gray shading is the rough endoplasmic reticulum, or the ER. Now this part is rough, and this part of the ER is smooth. So there's smooth ER and rough ER. What makes the ER appear rough under an electron microscope are all of these little ribosomes that have been attached to it. Yeah? So the little orange dots are the ribosomes, and you remember that that's where proteins are made, is on the ribosome. So there's a dance that occurs with the ribosome, the ER, and the amino acids and all the rest of it to make the protein. But I just said that proteins for export are made in the ER. So this is kind of what happens to make a protein. And we will spend a lot of time on this later in the course. But for right now, just try to follow along. Don't worry about all these details because they're not all going to be, you're not responsible for this whole story now. But I think it helps to tie it together, what's going on. So back to this figure, there's a small subunit, the large subunit, and this is a piece of RNA. And RNA is needed to make proteins. So what we have is that the RNA fits in between these two ribosomes, and then we start building a protein based on the instructions in the RNA. Okay, so all you have to remember is that this is going to build a protein. Okay, so take the ribosome now. This one is floating out in the cytoplasm of the cell. These ribosomes are not bound to the ER. They're free floating in the cytoplasm. The proteins that they make will stay in the cytoplasm. They're not going to be exported out of the cell. Whatever the proteins are that are made, they'll stay inside the cell and do whatever they're supposed to do. Okay. Back to this figure. This is the basic story of what's going on. So get yourself oriented. Scoot this up a bit. This is outside the cell. This is the extracellular side. Here is our phospholipid cell membrane. And here are some proteins that are embedded in it. Here's a vesicle being exocytosed. And so what we're going to do is follow this protein from when it was made, how it passes through the internal organelles to the outside. So back up here to the RER, ribosomes, and you know that ribosomes make proteins. So the protein gets made on the RER, the rough endoplasmic reticulum, and a piece of the endoplasmic reticulum buds off. It's like it pinches off a little piece of the cell membrane, and here is that little vesicle. It's a piece of the rough endoplasmic reticulum that has the green protein that was just made, and it carries it along. And then this moves to the next organelle, to the Golgi, and that green protein then moves through the Golgi, and as it moves through, it gets modified again. And you notice that there's a little red thing developing on the green protein. You may have to enlarge this or get your magnifying glass. Let me see if this works. Oh, there we go. That's better. So this is just the synthesis of the green protein. It gets modified as it moves now through the Golgi. Finally, a piece of the Golgi, which is also membrane bound, we'll talk about that next, buds off. So this is a piece of the Golgi membrane with the protein still attached. And then this vesicle, the orange vesicle, moves to the cell membrane and fuses with the cell membrane. Oh shoot, I'm off the film again here. There we go. So here is, backing up, here's the little protein that's been modified with a red star on it. It's moved through the Golgi. A piece of the Golgi buds off with the green protein stuck in the membrane. This vesicle fuses with the cell membrane. And there's the orange membrane of the Golgi fusing with the green cell membrane of the cell. And as these spread apart, there's the protein now stuck in the cell membrane. That's kind of neat. That's how proteins get built and stuck in the membrane of the cell. If you can just follow along with a basic sequence of events, that would be great. That's how, again, how proteins are made and inserted into the cell membrane. All proteins are made inside the cell on ribosomes. Okay. So I pretty much go back to our sheet and mention the Golgi. I just showed you pictures of it. Golgi body, Golgi apparatus, or just the Golgi. And this is the site of further protein modification. For further protein modification of exported proteins. Those are going to live outside the cell or stuck in the membrane. Okay, let's talk about the smooth endoplasmic reticulum. Again, refer back to the figure that's in your text. And the smooth ER. So again, this is structure function. These are the structures, and we're just trying to get a basic idea of their function. What do they do? Well, the smooth ER is involved with some lipid synthesis. So site of lipid synthesis. Phospholipids, other lipids are made in the smooth endoplasmic reticulum. Certain chemicals, certain drugs are detoxified. In the smooth ER. So cells that do a lot of detoxifying of drugs and chemicals, as you might expect, they have a lot of smooth ER. So liver cells, for example, that are detoxifying compounds from the blood, they have an extensive smooth ER inside their cells. Other cells that don't do so much drug detoxifying or aren't making that many lipids, their smooth ER is reduced. The smooth ER can also store calcium. So So stores calcium ions inside the smoothie art. We'll talk about that a good deal next quarter when we look at how muscle cells store calcium. And I think that's it for right now. If you would like, I would recommend a Khan Academy video. It was on protein synthesis, if I'm spelling Khan Academy right. I will look that back up and give you a link on it, because there's a lot more on protein synthesis that you may not want to look at right now. But it had a discussion of the ER and the Golgi. So I'm going to take a break for a moment, and when I come back with the next video, I will give you a link. So watch for link to that Khan Academy. I'll do the browsing and find one for you, and when we come back, we'll start with lysosomes, and then we'll do mitochondria, cytoskeleton, and we're pretty much done at that point. So, later.